Hello again with biochemistry section lectures. Welcome back. I'm Ms. Gona K, your lecturer in biochemistry. Today in our lecture we will talk about the DNA. We will learn during this lecture about the DNA, its own structure, its own functions, its history, the shape of DNA. Also we will learn about the transcription of DNA to RNA, also the translation of RNA to protein, how protein produce, what is the main function of it. We will learn about new terms such as genes, and we will learn about the codon table. First of all, where is DNA located? But before that, we know what is DNA. DNA is a simple name or abbreviation of deoxyribonucleic acids, which is a macromolecule known as nucleic acids. It has a double helix composed of a long strand of sugar and phosphate groups and also nitrogen bases. Usually the nitrogen bases in DNA are adenine, thymine, where they are binding to each other by a double bond, and also guanine and cytosine, where they are paired to each other by a triple bond, 1 sigma bond and 2 pi bonds. DNA organized into a structure called chromosomes. Its own location, it housed in the nucleus of the cell. Okay. Experiments in 1915 shows that the DNA has a hereditary materials. Scientists try to make more experiments to know the structure of the DNA. Until 1953, Watson and Crick proposed that the DNA has a double helix. This is the main structure of the DNA. This is the ribose base, and this is the phosphate. This is the deoxyribose. Actually, we call it a deoxyribonucleic acid because here we have one oxygen which is not present. We have only hydrogen. That's why it's called deoxy. This ribose base has one oxygen less. That's why they called a deoxyribose nucleic acids. And these are the nitrogen bases, where some of them has double cyclic, which is called purine bases, such as adenine and guanine. The one have one cycle called pyrimidine, which they are cytosine and thymine. Please note the name and which one is the purine and which one is pyrimidine bases. In RNA, we have a ribose sugar base with phosphate, and we have uracil instead of thymine. These are the nitrogen bases structure, the adenine, guanine, thymine, and cytosine. Let's now move to another term, which is gene. What is gene? Gene is the unit of hereditary. Each gene is responsible for a single inherited property or a characteristic property in the organisms, such as phenotypes. Since cells all need to be complete sets of genes, it is necessary for them, the original cell, to duplicate it into its own genes before it's divided. Then after division, we have two processes, either mitosis or meiosis. The cell division will undergoes through it via 3 phases. The first phase is called a prophase. The second one is metaphase. The third one, which is the final one from the parental cell, goes through it until it gets the daughter cell called the anaphase. What is the difference between meiosis and mitosis? As it's shown here and as it's obvious, the main difference between them, that in the mitosis we have father and mother, two chromosomes, sisters. But here we have the chromosomes as a father and mother, they are duplicated. Here we see the results of the daughter cell as it's shown. In the mitosis will be 2n, while the daughter cell result in the final step in meiosis in anaphase and telophase will be 1n. Although we have in the third phase we have only one cell division in the mitosis, while in meiosis we have one extra step to get the daughter cell. So this one is 2 and this one only one phase. Moreover, which is interesting to know for me, to you, the meiosis usually occur in the only sexual cell division, while the mitosis occurs in all somatic, such as the body cells, the non-sexual one, such as when you need to make RBCs, the non-somatic processes inside the development, and etc. Genes are made of DNA and make up to the chromosomes. Each chromosome must be accurately copied. Upon the cell division, both daughter cells will receive identical set of chromosomes. Let's now move to how the DNA packing itself in the eukaryotic cells. The DNA starts folding by coiling itself around histones, which they have a positively charged proteins. The histones here, as it shows here, we have 8 histones that the DNA wrapping itself around it. As I mentioned, it has a positively charged. Does that mean that the DNA might have a negative charge? From where it's get it? As we mentioned, the main structure of DNA, they have a phosphate groups. The phosphate group might give it the negative charge. The negative charge comes from it to be neutralized. Each 200 base pair of these of DNA wrapped around 9 histones. As it's shown here, we have 8 histones, and the 9th one, usually protein H1, make the knob of the wrapping and make it to connect to each other and show the double helix structures. 8 histone closer together, which has a 140 base pair of DNA coiled around them, a core particle called nucleosomes. This is only 2 nanometer DNA. What is the thickness of the this coil, which is wrapped each other around it? Each coil, which is wrapped, has 30 nanometer fiber diameter loop, which is approximately this one 300 nanometer long for this a very short DNA. As it's shown here, inside the chromosomes, how it's wrapped itself, the DNA has a double helix. It's wrapped its own self around the histones and strength with and bend each others by 9 to 11 nanometers. The chromosomes will be, the nucleosomes will be folded around each other and supercoiled around each other in a fibers with a diameter of 30 nanometer, as I told you, around each other. Then it will packed in the chromatids. Here, as it's shown in the structure of the DNA, we have the 9 histones and the 8 histones, and the 1 histones that make the knob as a linker, which is work as a linker to make the DNA and the chromatosomes rapid with each others. Alright, a linker regions, which is the ninth one, consisting of the remaining 60 base pair of the DNA and the ninth histones molecule join each core particle to the next and make this kind of shape. The chain of the nucleosomes would be helically into a giant polynucleotide structure with the 6 nucleosomes per turn, the 13 nanometer fiber. Here, as it's shown, in turn with it, the fiber are looped back and forth, back and forth. There are about 50 of the solenoidal turn per loop, and the end of the loops are attached to the protein scaffold. This is the structure of DNA as it's shown. 18 of these loops will be wounded around each other radially around the chromosomes axis to give a mini band to make itself has the most kind of base pairs and in smallest place. Roughly 1 million such as mini bands will be formed to each other to form the little bit of duplicated place of in the chromosomes. Now let we talk about the RNA replication. DNA replication. How DNA replicates its own self. What we mean by replication? It means DNA copying itself in order to pass the genetic information onto newly formed daughter cell. DNA must be unwounded first because it has its own double helical structure. So first of all, it must be unwounded to allow the cell replicate machinery to copy each strand. Each replicated molecule is composed of a strand from the original DNA molecule, newly formed strand. Each parent strand remains intact to each others. Every DNA molecule is a half old, half new. The new one, the daughter cell, will be half old and half new DNA. The replication process generally identical in DNA molecules. DNA replicate occur in intraphase, a stage prior to start the division